your time with respect to 12 minutes is up, I will stop you then and there. I won't care if you are on slide 2, if you're on slide 15, if you have just a conclusion slide left. I don't care. Okay, if there is a time, you have to abide by it. Okay, so my advice to you is, as soon as you hear the warning, please start wrapping up. Even if you have some results left to discuss, it is always better to end with a conclusion than end abruptly on the results. Okay, with that said, after that, we will either have a Q&A session. That is, I will ask you some questions regards to your approach, what all stuff you guys did, what all analysis you guys performed, or I will give you some suggestions or advice from my end. Okay, it will be an open-ended discussion, so I expect you guys to also answer. Okay, this time it won't be thumbs up and thumbs downs, it will be answers. Okay, I want you all to unmute and answer. Okay, and the questions or the discussion need not be answered by only the presenter. I want the entire group to participate in the discussion. Is that clear with everybody? Can I please get a thumbs up if that is clear? Okay, Bhavni, we shall begin.
Okay. Akshay, please start. I will be starting the timer. Suryadev, hope you can see the timer at the top right corner of the screen. I haven't
Not yet.
started, but once
Yes.
I started, you will be able to see them.
Okay. Okay.
Yeah. Akshay, please change it to slideshow.
Yes, ma'am.
And all the very best, guys.
All the best, everybody. All the best, group one.
Thank you.
Yeah.
I hope my screen is visible.
Yes. Screen is visible, and I'm starting the timer now. Yeah, let's begin.
So a very good evening to everyone. We are Group 1. I'm Akshay, and I'm here to present my group's mini project titled Machine Learning Insights into Genes Involved in Type 2 Diabetes. So for our project, we use the gene expression data that was given to us to distinguish between patients with type 2 diabetes and against healthy control subjects. So we applied multiple machine learning models and we compared their performances. Apart from that, we also identified the genes that contributed majorly in helping shape the model's prediction of whether an individual has type 2 diabetes or not. To give you an overview of the dataset that we've used, it's a gene expression dataset that gives us a comparison between type 2 diabetes patients and healthy control subjects. This is a problem of binary classification wherein there are only two outcomes possible, either the patient is diabetic or non-diabetic, or in simpler terms, a comparison between condition and control. This data set is also high dimensional, meaning that the number of genes for which the expression data was given, or the number of features basically that we've used, is greater than the number of samples itself. So that makes this data set high dimensional. And also another feature of this data set is that it's balanced between the two classes, diabetic and non-diabetic. This helps us to remove any bias that might occur during model training. So for our project, we use 3 different machine learning models, namely SVM, Decision Tree, and XGBoost. The support vector machine model, or the SVM model, is a linear classifier, which for us, we considered as a baseline model. Our next best model was a decision tree, which makes a prediction based on simple decision rules, and it's easy to interpret. Our third machine ML model was XGBoost, which is basically an ensemble approach where it combines multiple decision trees to arrive at a prediction. Our motive behind choosing these 3 models was to compare between linear, tree-based, and the ensemble approaches. Next, to analyze how well each model performed, we considered 4 parameters or 4 evaluation metrics, namely accuracy, precision, F1 score, and the AUC score. Here, accuracy refers to how many of the predictions that the models made are actually correct. Precision is of the number of positive predictions that the models gave us, how many of them are actually positive. For example, if the model said that 10 patients are diabetic, we see that how many of these patients are actually diabetic. That is how well the model made a true prediction. And then talking about F1 score, it's a balance between precision and recall. Recall is slightly different from precision, as in when we talk about recall, we see that of the number of actually diabetic patients, how many of them was were the models able to detect? And then talking about the AUC score, it's a direct reflection of how well a model is able to distinguish between the two classes. Here is a summary table mentioning the values of the four evaluation metrics that we just talked about. We can see that the SVM model acts as the baseline model with moderate scores. We have slightly better values for the decision tree, which we consider as the best model for us. And then here we can see that the XGBoost model has a score of 1 for all the four metrics. This, however, is considered overfitting, and I'll be talking about why XGBoost model is considered as an overfitting model for us on a separate slide later in the presentation. Here are just images of the results that we've got for the evaluation metrics for the SVM model, the decision tree model, and the XGBoost model. Now let me talk about the AUC scores. We got an AUC score of 0.79 for the SVM model, which suggests moderate discrimination between the two classes. Then we got a slightly better score, 0.94 for the decision tree, which indicates that the separation of classes was slightly better for this. And like we've seen in the table earlier, XGBoost got an AUC score of 1. This achieves perfect separation, but this suggests overfitting. Here are the ROC curves for each of the models. Here, the diagonal dotted line in the middle refers to the random guessing line, and the closer our curve is to the model, the weaker its discriminating capacity is. So we can see that the curve moves slightly farther away as we move from the SVM model to the decision tree model and then to XGBoost. And then another aspect that we've considered was the confusion matrix, which basically gives us at a glance the number of positive and negative predictions. So it talks about the true positive and negative values and the false positive and negative values. We saw that the SVM model misclassified a significant number of diabetic samples. Decision tree was slightly better by reducing the number of false positives and false negatives. And then for XGBoost, again, there was zero misclassification, which again suggests overfitting. So here is the confusion matrix for the SVM model, wherein the rows depict true label, as in if the patient really is diabetic or not, and the columns represent what the model predicted. So here, the values 83 and 33 refer to the true negative and true positive values, respectively. So we can see that these two predictions were correct given by the SVM model. However, 17 and 21 are the false positive and false negative values given by the model, respectively. And these are kind of what made the SVM model get slightly lower values, slightly lower precision or AUC values compared to the other models. Then we can see that the values have reduced for decision tree. The false positive and false negative values especially have reduced a lot to 1 and 2. And then for XGBoost, we see that the false positives and false negatives are entirely 0, and that there were 24 positive predictions and that 24 true negative and true positive values. So this looks ideal for XGBoost, but again, it's overfitting. Now, let me talk about the genes that shape the decision tree's ability to predict the disease correctly. So the most influential gene to shape the decision tree's performance was the PPARG gene, followed by the TCF7L2 and then CEP170 genes. These three genes in this order were what helped the decision tree make the predictions that it made. Here is a plot that suggests the same, where we can see for PPARG we have the longest bar, meaning it was the most significant gene for decision tree, followed by TCF7L2 and then CEP170. This is the features important plot for the SVM model, where we can see that this model heavily relied on the glucose levels to make its decisions, followed by features like BMI, diabetes pedigree function, pregnancies, age, etc. And this is the features important plot for XGBoost model, where again, the model highly relied on the PPARG gene, similar to the decision tree model, to make its predictions, followed by TCF7L2. The two different genes here as compared to decision tree are GCK and then IRS1. Now, let me talk about the biological interpretation of these genes, key genes that we've identified for all the models. So, PPARG gene is responsible for regulating insulin sensitivity and adipocyte differentiation. It basically controls how body stores fat and how it responds to insulin, especially in adipose tissues. And the second most influential gene is TCF7L2, which is involved in the WNT signaling pathway, and it majorly controls glucose homeostasis or the balance of glucose levels in the body. Then we have glucokinase GCK, which is very important in pathways related to glucose metabolism. And then we have IRS1, which is insulin receptor substrate 1, that is majorly involved in the insulin signaling pathway. We also referred to several papers that have been cited at the end of the presentation to get this information, as in identify which pathways are each of these genes involved in that control or that regulate the condition of type 2 diabetes in patients. So PPARG, as I mentioned before, is involved in insulin sensitivity, and that is controlled by the PPAR signaling pathway. The TCF gene is involved in WNT pathway, where this pathway influences how pancreatic cells produce insulin in response to the levels of glucose present in the body at the moment. Then glucokinase is majorly involved in glycolysis pathway and other glucose metabolism pathways. This also regulates
You have
the
2 minutes
production of.
left.
I'm sorry, sir.
You have 2 minutes left. Please continue.
Thank you. GCK acts as a glucose sensor, and that also regulates insulin release based on the glucose levels. And then finally, IRS1 is involved in the insulin signaling pathway, where after insulin binds to the receptor cells, IRS1 helps to transmit this signal into the inside of the cells. Now, as I mentioned before, now I'll talk about why the XGBoost model is considered as overfitting. So the data set that we've used is a synthetic data set that basically is high dimensional, as I mentioned at the beginning of the presentation. So because of it being high dimensional, it increases the overfitting risk. So seeing these values of 1 for all the parameters is expected when we are dealing with such a synthetic data set. However, it's uncommon for biological data. That point is to be noted. Now, talking about the limitations that we faced throughout the length of this project, XGBoost showed signs of overfitting, and then our data set was high dimensional, which increases the risk of overfitting, and it didn't correctly train the patterns. Instead, it focused on the noise because of the because of the large data set. There's also no external validation of the data set, and also the key genes that we've identified, even those we've inferred from the model's predictions that we've got and not experimentally. So these were some of the limitations. In conclusion, SVM was the baseline model, which gave us moderate values. Decision tree gave us clean values with good interpretability, and then XGBoost gave us excellent results. However, it showed signs of overfitting. So overall, the study talked about this project that we performed. It talked about how we can combine machine learning and biology to talk about diabetes-related genes. That brings me to the end of my presentation. Thank you very much.
Thank you very much, Group 1. Excellent presentation. I love the fact that you guys had a very nice structure to it. You guys stated clearly what you were trying to do, what all metrics you're trying to use, what all models you're using. You gave, I felt, with the very basic and limited knowledge that you guys have gathered throughout this week, you guys gave very good conclusions as to why you are choosing what model. Okay. One piece of suggestion I have for you guys is that references at the end is not really the best way to go. I would prefer if when you are talking about the explanation as to why each gene is so important in this biological scenario, you should put the reference of that paper below that slide itself. Okay. So
Yes.
that is one suggestion that I have for you guys. So apart from that, a few questions I have. First thing, can you please take me back to the feature importance plot of SVM?
Yes.
Yeah. So is this feature importance plot from the gene expression data that I gave you guys?
Yes, sir.
Uh, why are the features then glucose, BMI, diabetes pedigree function, pregnancies, age, blood pressure? Why are these the features? This is the diabetes data set that we use during class, right? Who did this SVM model in your group? Can you please check with them if they actually performed it on the gene expression data set?
Yes, sir. Let me calculate.
No, so, I mean, was there a confusion here? Because for XGBoost and decision trees, I believe you guys used the gene expression data set that was shared. What happened with SVM? Anybody from group one? Siddharth?
Yeah, good evening,
Uday?
sir.
Yeah,
I'm the,
Uday. Yeah, tell me.
I have done the SBM, sir. While placing, it might be in the confusion. I have given two plots, sir. I have for the sharp plot. For actually SVM, the sharp plot will be it cannot be done. So I have converted the data into linear data and I performed data. I have and I have given two plots to them.
Uh-huh.
So it was the linear SVM plot.
Yeah, so
So
you
the
will
before
get
one
a
would be correct, sir.
before one in the sense which one?
I have given. Can I place it in the Telegram group?
Yeah, sure. Go ahead. Put it in Telegram.
Okay, sir. 2 minutes.
Yeah. Okay, but you did do the feature importance, right? SVM, you obviously, I mean, it's not that you cannot do SHAP on it. You need not. SVM already has its own feature importance module. But yeah,
Okay.
I hope you have actually done that, right?
Yeah, I have done it.
Okay, okay. No worries, no worries. Okay, so my question to you, Uday, then specifically because you are the one who worked on SVM, is which kernel did you use for your SVM model and why?
It's a linear kernel, sir.
Okay, you use the linear kernel. Okay, good enough. And why did you think of using the linear kernel in this scenario?
It's a straight line separation when the data can be separated linearly. I thought the data
Mhm.
can be separated linearly, so I used
Mhm.
the linear kernel.
Okay,
And if I go with the RBF for a polynomial, it would be curved or a polynomial curve. So I
okay.
thought that the data can be separated linearly.
Okay, no worries. I think a better and a more objective reason here would be that right now, your the what was the goal with the SVM? The goal with the SVM was to form a baseline, right? So something that just does the job, not too good, not too bad, right? That is the idea of a baseline, right? Uday?
Yes. Yes.
Yeah.
Yes.
So I think that could be a better and more objective reason that the baseline could be if the baseline is something very simple, it always helps in both interpretation as well as when you are making improvements you can get to know better as to why you are improving because it is better than your baseline. Okay, so I believe that could be a better reason with respect to why you use the linear kernel. That is fine. So similarly, Uday, you could have also used logistic regression, right? Because this is something we discussed, that SVM with a linear kernel on a classification problem effectively performs logistic regression. Did you try logistic regression?
No,
Did
sir.
anybody try? Okay. Try it once. Maybe you'll get the similar results. Okay.
Okay.
Just try it and experiment once. Maybe. Just
Okay.
maybe you'll get similar results. Okay.
Sure. Sir.
Um, another thing is none of the metrics that I saw were expressed as mean plus minus standard deviation. So did
Yes,
you
sir.
guys perform,
Accuracy. You have mentioned it like to
was it?
accuracy score. You have mentioned already like to present the accuracy score in standard deviation.
So this is not mentioned as Mean plus minus standard deviation. Where is the standard deviation here? I don't see it. Do you guys see it?
No, no, sir.
Yeah. So did you guys do cross validation for any of the models? Group 1. Anybody? Was cross validation performed? Guys, come on, don't be silent. If it was not performed, just say that, okay, we did not do cross. Or if you're not sure, just say that, okay, we are not sure.
We are not sure.
Okay, you're not sure. Okay. No worries. So my advice then in this regard is, if you have not, then please do perform cross validation. That is your biggest weapon that can help you prevent any overfitting problems. Okay, the whole point of cross validation is that where you bootstrap data and you train it on different data sets, right? From the same data set, you create different data sets, you train it on different data set, test it on different data set, so that you don't get something, you don't overestimate or underestimate your model. Okay, so do perform cross validation, and always, and this is for everybody, showcase your metrics in a mean plus minus standard deviation format. Okay, and mention how many folds you have done for your cross validation. Okay, now Akshaya, if you can take me to your confusion matrix for the SVM, can you please take me there?
Yes.
Yeah, a very simple question, anybody of you can answer. Which box here is the true positive and which box is the true negative?
The box with 83, the top
83
left
is
one
a
is
true
true.
positive.
Is it the true positive? Are you sure?
It's the true. It's the true negatives.
And why do you say it is the true negative?
Because the rows here is the true label. So the number of patients who are actually non-diabetic, and then the first row is what the model predicted.
Correct.
So yeah.
So basically, correct me if I'm wrong, you're trying to say is that both the prediction as well as the true label is 0 or the negative class for this, which is 83, right? That's what
Yes.
you're trying to say, right? Therefore, these are true negatives, right? Or whatever the model has predicted as negative, and they are also truly negative, correct?
Right. Yes.
Is that how the encoding was also performed? I believe that is how it was performed. Can anybody
Yes.
confirm? Yes. Did you need to do any encoding on your data, or was the label already encoded? Do you guys understand the question?
Yes, sir, we do.
So tell me here, was the target label already encoded or did you need to do any encoding? Okay, tell me very simply. Did the target label have diabetes healthy like that written in text, or was it 0 and 1?
It was 0 and 1, sir.
Okay. So now answer me the previous question.
It's already encoded.
It is already encoded. Okay, that is what I wanted to know. Okay, so I hope you guys have actually gone through the data before blindly jumping into the analysis. Okay, because these are questions which should be at the tip of your tongue if you have actually performed the analysis on your own. Okay, it is absolutely fine if you have borrowed code from other sources. That is absolutely fine, but please do understand the analysis you are doing, because otherwise there is no point to this entire thing. Okay. Okay, that is it for group 1 then. Overall, a very good job done, a very really good job done. I'm really impressed with you guys. Please keep in mind the suggestions that I gave you, and all the best for your future endeavors with machine learning and biology. Okay, thank you so much, group 1.
Thank you, sir.
Thank you, Suryadev, and thank you, Group 1. Group 2, Varshini,
ma'am.
yeah, please start your presentation.
Once again, I'm trying to share my screen. Just let me know if it's visible.
Just
Fine.
before group 2 begins, just a second, Bhavani. So,
Yeah.
group 1, as far as all the members of your group are concerned, did all of them contribute equally? Or was it like a 2, 3 people working? Can you please confirm?
No, sir, we divided the work equally, and if one of us were facing problems, we made sure that the
Okay.
others helped.
So all of you work together, right? Okay. Okay. Thank you, group
Yes.
1.
Can I
Varshini,
start, sir?
yes. Suryadeep, shall we begin with?
Yes, let's begin group 2.
All
All right.
Group 2, please start.
Yeah, sure, ma'am. Thank you. Good evening, everyone. We are from Group 2, and I'm Varshini. I'll be presenting a project on Alzheimer's disease prediction using gene expression data and machine learning. Uh, so, yeah, the problem statement here we are trying to address is that Alzheimer's disease is a progressive neurogenetic disorder that primarily affects our memory and cognitive function. The major challenge is that early symptoms often overlap with normal aging, making diagnosis difficult. So, by combining gene expression and analysis with machine learning, we can uncover the hidden molecular patterns that are easily detectable using traditional methods. So, we tried addressing our goals in twofold. So, for the first one is to classify Alzheimer's and healthy control samples. Second one is to identify genes that play a significant role in the disease prediction. Moving on to the data set consists of gene expression profiles collected from individuals which are diagnosed by who are diagnosed by Alzheimer's disease and also from the healthy control subjects. Each sample represents one individual and a feature corresponds to the expression level of a specific gene. Since gene expression values are numerical and high dimensional, this data set is ideal for machine learning based pattern recognitions. Now, the data set contains 216 samples with a very high dimensionality of 19,000 to 98 genes. The target variable is the class variable indicating Alzheimer's or healthy control. Importantly, the data set is balanced with the 108 samples and 108 healthy samples, like disease and healthy. This balance helps us ensure unbiased training and fair evaluation. Such data sets, they have applications in Alzheimer prediction, biomarker discovery, and gene importance analysis, and also used for medical decision support systems. Here we have used 3 models, random forest, support vector machine, and XGBoost. The random forest, this ensemble is an ensemble model that combines multiple decision trees to reduce overfitting, whereas SVM, as the name suggests, it works by finding an optimal hyperplane that maximizes the margin between classes and also perform very well in high dimensional spaces. XGBoost is a boosting algorithm that builds trees sequentially, where each model corrects errors from the previous one, making it more powerful. The evaluation matrices we used here are accuracy, precision, F1 score, confusion matrix, and ROC curve, you see. Accuracy, it measures overall correctness, while precision will tell how many predicted Alzheimer cases were actually correct. F1, it balances the performance, especially this is used in medical data sets, where the AUC ROC curve, they understand the classification behavior across different thresholds. So yeah, this shows the comparison table. So now we can see that SVM achieved an accuracy about 84%. And coming to random forest, 81%, and XGBoost, it has showed a perfect performance. However, such perfect accuracy in biological data often raises concerns about overfitting, which I'll be discussing later. Now, the four pillars of accuracy are true positive, false negatives, and true negatives. From this, and this basically the true positives and true negatives represent correct predictions, while as the false positives and false negatives represent the error. In medical application, false negatives are especially critical because a missing disease or diagnosis can have serious consequences. So yeah, starting with the random forest, this is a supervised learning algorithm used for both classification and regression. Instead of relying on a single decision tree, it builds many trees using random subsets of data and features. Each tree gives a prediction, and the final output is based on the majority voting, which improves the robustness. The random forest confusion matrix here, as said, it is achieved
it achieved 81.8% accuracy. The dominant diagonal in the confusion, it indicates high number of correct predictions. The model performs well in identifying healthy controls, though a few Alzheimer cases are missed, suggesting a room for improvement and sensitivity. Coming to the ROC-AUC curve, it shows a AUC value of 0.91. This indicates strong discriminatory power and the model's ability to effectively separate Alzheimer's samples from the controls. Now these are some important genes that we have taken. This plot, it shows the top genes influencing random forest predictions. Genes like APOE and APP, they have highest importance, which aligns with the known biological evidence related to Alzheimer's disease. APOE is the gene that strongly increases the risk of Alzheimer's disease, whereas APP, it is the gene that produces amyloid protein, which is harmful for plaques in Alzheimer's. So amyloid protein is abnormal protein that clumps together in the brain and is strongly linked to memory loss. And then moving on to SVM. Here, SVM support vector machine is a supervised learning algorithm that focuses on finding the best boundary between the classes. It selects a hyperplane, right? And then maximizes the distance from the closest data points, called support vectors. As the name said, the support vectors play a crucial role in defining the decision boundary. Coming to the confusion matrix, it is the SVM model achieved around 85.1% with very few missed positive cases. Although some false positives were present, SVM showed a strong sensitivity, which is desirable for early diagnosis detection. And the RC AUC curve, right? This shows an AUC of 0.91, indicates a good balance between sensitivity and specificity, confirming reliable classification performance. The feature import and the confusion matrix, right? Yeah, this achieved around 85%. Yeah, ROC curve and the feature importance. Here we see the top genes identified by SVM. This is based on the coefficient values. The genes like APP, as said, amyloid protein, PSEN1 and PSEN2. These both are, they control how amyloid protein is made. And yeah, the XE-boost. This stands for extreme gradient boosting. Unlike random forest, which builds trees independently, XE-boost builds trees sequentially. Each new tree focuses on correcting the mistakes of the previous one, making the model extremely powerful. The confusion matrix, this model, it achieved 100% accuracy with no misclassifications. The perfect diagonal indicates complete separation between Alzheimer's and the control samples, which is unusual in biological data. The RSC curve, this shows an AUC of 1.0, indicating perfect discrimination. While impressive, this result, it raises concern about generalization and potential overfitting. The XE-boost feature buttons, as said, APP genes controls the amyloid protein. And yeah, APP, and it appears to be the most influential gene in XE-boost, followed by APOE and then PSEN1. This suggests the model relies heavily on known Alzheimer's biomarkers along with additional supporting genes. So yeah, coming to the overfitting analysis, perfect accuracy is often a sign of overfitting, and especially in high-dimensional biological data with nearly around 19,298 genes precisely, and only 216 sample, XE-boost may be memorizing the patterns instead of learning generalizable relationships. So this shows the comparison of the ROC-AUC curve for the three models. And yeah, SVM and random forest achieved a AUC of 0.91, and XE-boost shows a perfect performance, which further supports the idea that it may be overfitting rather than genuinely outperforming the other models. These are the sharp plots. This first one is of random forest, and the next one is SVM, and the last one is of XE-boost. The sharp plots, they basically interpret how model predictions by showing how individual genes influence the classification. They provide transparency and explainability, which is essential in biomedical machine learning applications. Coming to the conclusion, in conclusion, we successfully applied all the machine learning models to classify Alzheimer's disease using gene expression data. SVM had provided strong baseline performance, random forest captured nonlinear relationships and gene importance, while XE-boost, it showed extreme performance with possible overfitting. So overall, SVM and random forest appear more reliable for real-world biological data. And Yeah, these were the some research papers that we went for Alzheimer gene expression studies and also machine learning algorithms. So yeah, that's bring us to the end of our presentation, and we are happy to take any questions. Thank you.
Thank you so much, Group 2. That was a lovely presentation. Very well done. So again, a very similar thing with Group 1 here. I noticed that you guys also have not mentioned your metrics with respect to the mean plus minus standard deviation. So let me ask you this. Did you guys do any form of cross validation?
Uh, no, sir, we did not. We're not sure.
Okay. If you have not, then my suggestion is please revisit this and do cross validation. Maybe with XGBoost after cross validation, you might not see accuracies of one. You might
Uh,
see something less, and that might actually debunk some myth that you have that it is overfitting. I'm not saying that you will definitely get it, but there is a possibility. There is always a possibility. Okay.
definitely, sir. We'll take that into
Okay.
consideration.
Um, apart from that, I wanted to ask for your random forest or your XGBoost model, did you guys do any form of regularization? As in, did you guys try different values of gamma, different values of the pruning parameters, different values of lambda? Was that tried?
Sorry to interrupt, sir, but apparently we have done, cross validation for XGBoost.
Okay, you did cross validation for
Yeah,
XGBoost,
only for
and
XGBoost.
okay, only for XGBoost. Okay.
Yeah,
Okay, no worries. If you have done cross validation and still you got one, okay, that is absolutely valid since these are synthetic data.
yeah.
That is fine. But please then always mention your results in the form of mean plus minus standard deviation. Okay.
Sure, sir.
And so how many folds did you do for cross validation?
So,
How many folds for cross validation? For how many
I
folds? Whoever is telling you the
mm,
answer, they can just unmute and answer, right? Whoever is in your group.
20, I guess, sir, if I'm not wrong.
20 fold. Okay.
Yeah,
Okay.
it's Wahaj who did the XGBoost. So.
Okay. Wahaj, if you're part of, is he part of the call
Sir,
today?
yes, sir. I
Yeah,
am
yeah,
part
he
of the
is.
call. I'm actually outside, sir. Sir,
Ah,
I've
okay.
taken the help of this thing. So in that, I got first, I got these overfitting plots, sir. Then I
Uh-huh.
tried doing that, and I got a different plot, but I was not sure if I was in the right direction. So we
Okay.
used these in the presentation. So
Okay, that is absolutely fine. Let me
Yeah.
ask you this, Wahaj. Did you use any regularization parameters, any pruning parameters like gamma, lambda? Did
no,
you
sir,
try with
I
different
don't think so,
values?
sir. No, sir.
Okay, okay. So my suggestion to you guys is sometimes it does lie with you guys that you can actually tweak these parameters and help prevent overfitting and make these models more generalizable. Okay. So maybe try that sometime. Okay.
Yes, sir.
I'm not
I'll
saying
try
it will
doing it, sir.
definitely
Yes.
work, but with real life biological data sets, that is something we often end up doing. So that is one of the advices that I have for you. And Varshini, if you can take me to the SHAP plots once. I have one question from the SHAP plots. Yeah, just make it full screen so that I can also
Oh,
see
yeah,
it.
1 second, sir.
Yeah. So the one on the one in the middle,
This
right?
one, yeah.
This is from Random Forest, right?
No, this is from SVM.
Okay, this is from SVM. Perfect. Varshini, either you or anybody from your group, I want any one of you to explain this SHAP plot in terms of the expression of APP and PESEN1,
Any plot, sir, or the one in the
the
middle
middle
one?
one, the one in the middle. What do you understand from this plot? I want to know.
Mm,
Anybody from the group can jump in,
So can
guys.
I
Yes,
explain?
please.
Yes.
Yes, absolutely.
So the y-axis here represents the genes that we used, and the x-axis is the SHAP value.
Mm-hmm.
Right side, it pushes the predictions towards the diseases, whereas left, it shows towards the control.
Okay.
And each dot here is one sample,
Mm-hmm.
and the pink color dot shows high gene expression, whereas the blue color dot the blue dots show low gene expression.
Mm-hmm.
And this for uh, if also
So,
this
so, so what does it mean for APP? If
Can
blue
I add
means
that?
yes,
Yeah,
Varshini, go ahead.
yeah, yeah. So APP here, as it's on the top and it has like strong influence. And
Mm-hmm.
here, higher red, this higher APP expression, this
Mm-hmm.
pushes towards the model, and the lower, the blueprint one, it's towards the
Mm-hmm.
healthy control, like not diseased. So it's directly involved. The biological link is that it's directly involved in amyloid beta plaque formation.
Are you sure what you just said? Is it that?
Mm,
So,
yeah,
what is the left side of the shaft value axis? The x-axis. What is the left side? Left side is the negative side, right?
yeah, it pushes us
So,
towards control.
yes. So, higher expression values of APP are more characteristic of the control or the disease here, which is Alzheimer's.
Yeah, sir.
No, no, what I asked you, which one is it? Higher expression values of APP. You just said that the pink ones are the higher expression values, right?
Yeah,
So higher expression value of APP is towards the left, correct?
yeah,
And the left typically represents the negative class or class 0, correct?
yeah, sir.
So higher expression values are associated more with the control or the Alzheimer's.
Alzheimer's?
Are you sure, Varshini? Think about it.
Well, I guess it's more towards the control cause negative to and it's towards zero, right?
Correct. Ananya, do you understand this? Would you agree?
Yes, sir.
Okay. So higher expression values of APP are more towards the control, whereas
Control.
lower expression values of APP are more towards Alzheimer's. So, what does that tell you? That tells you that APP is probably a gene which is significantly downregulated in the Alzheimer's condition, right? Does that make sense?
Yeah, sir.
This is exactly how you are going to use these sharp plots. These sharp plots are not for just pretty colors on your presentation, okay? You have
Yeah,
to
sir.
make sense of it and you have to figure out what it is trying to tell you, whether it is a down regulation, up regulation, with respect to what. You should all know this, okay?
Yes, sir.
So tell me, for PSEN2, what does it mean? We just did for APP. What does it mean for PSEN2? Pershini, you tell me.
So the left side one are for control and the right one are for the, deceased.
Okay, so high expression of PSEN2 associates more with control or Alzheimer's?
Control.
Control. And low expression with Alzheimer's, correct?
Alzheimer's.
So
Yeah.
PSEN2 would be technically upregulated or downregulated in Alzheimer's.
PSU
PSEN2, the second
down,
one,
sir.
downregulated.
Yeah.
Correct?
Yes, sir.
Okay. Ananya, Wahaj, and Nawaz, would you agree with what Varshini just said?
Yes,
Can you
sir.
please?
Yes, sir, I guess.
Okay. Okay. Thank you so much, Group 2. That's all from my side. Thank you so much
Yeah.
for this presentation. Very well done. Really happy with your presentation. And a round of applause for you guys. Thank you. Thank you.
Thank you
Thank you
so
so
much.
much, sir.
Thank you, Group 2, and thank you, Suryadev. Moving forward, Group 3. Vanaspani, please start your presentation. And Krishna, right? Krishna is the presenter. Krishna, are you ready?
Yes, ma'am. Yes, ma'am, I'm ready.
Yeah. And Vanaspani,
तो नहीं मंगाया कुछ भी सिर्फ जूस मंगाया और वो सॉसेज मंगाई
who is that? Can you please
मंगाई
unmute?
मैं खा लेता हूँ
Vahash, could you please unmute yourself?
Can I start, ma'am?
Give me a minute, please. Yeah. You can start.
Yeah, okay, thank you. Good evening, everyone. I am Krishna Karthik from Group 3, and today we are going to present on our project, which is machine learning based classification of rheumatoid arthritis using gene expression data. The goal is to check whether machine learning models can distinguish rheumatoid patients from healthy controls using gene level information. We are going to, okay, introduction. Rheumatoid arthritis is a chronic autoimmune disease that mainly affects joints and causes inflammation and pain. Traditional diagnosis can be slow, so gene expression data helps us understand molecular differences between rheumatoid patients and healthy individuals. In this project, we use machine learning models to classify RA and control samples based on gene expression patterns. Dataset description. The dataset contains 234 samples in total. Out of these, 117 are rheumatoid arthritis patients and 117 are healthy controls. Each sample has around 19,000 gene expression features, which makes this a high-dimensional dataset. The dataset is balanced on the so the models are not biased towards one class. Methodology. First, the data was loaded and pre-processed. Disease labels were encoded into numerical form. The data set was split into training and testing sets using an 80-20 split. Features scaling and apply Feature scaling was applied, and then multiple machine learning models were trained and evaluated using the performance matrix. And machine learning models used. So we used three machine learning models. One was logistic regression. Uh, Logistic regression was used as a baseline linear model, simple and computationally efficient for high dimensional data. It provided moderate accuracy and served as a reference for comparing advanced models. Then we used random forest classifier. Random forest classifier was chosen because it can handle complex and nonlinear relationships in gene data. This achieved the highest accuracy and ROC AUC score in this study. It enabled identification of biologically relevant genes through feature importance. Then we used support vector machine, which was also tested, but it struggled due to the extremely high number of features. It showed prediction bias without dimensionality reduction or tuning. It resulted in lower accuracy compared to other two models. Evaluation matrix. To evaluate model performance, we used accuracy, precision, recall, F1 score, ROC AUC, and confusion matrix. ROC AUC tells us how well the model separates RA patients and healthy controls. This is the table. This predicts the true positive, false positive predicted by the three matrices we used. Uh, as we can see, logistic regression, it predicted that 16 true negative, 16 true negative, like it predicted the accuracy was not that good in logistic regression, but and even in SVM it was not that good. But random forest gave us very accurate results compared to other two. Logistic regression results. Logistic regression achieved an accuracy of around 66% with an ROC AUC score of 0.66. The performance is moderate, indicated that RA and control samples are not linearly separable. Since this is a linear model, it struggles to capture complex gene interactions. It mainly serves as a baseline of comparison. Random Forest performed the best among all the models. It achieved an accuracy of about 85% and an ROC-AUC score of 0.97. This shows that nonlinear relationships among genes are very important in RA classification. Random Forest effectively handles high-dimensional data gene expression. Support vector machines showed poor performance with an accuracy of around 49% and a low ROC-AUC score. This is because SVM is sensitive to very high dimensional data without dimensionality reduction or tuning. As a result, it showed prediction bias towards one class. This shows the ROC-AUC curve analysis. This slide shows the ROC-AOC curves for all three models. The ROC-AOC curve plots truly positive rate against false positive rate. The random forest curve is closest to the top left corner, indicating excellent performance. Logistic regression shows moderate performance, while SVM performs poorly. This confirms the random forest is most reliable model. Feature important analysis. This bar plot shows the most top 10 most important genes identified by the random forest model. Genes like IL-1, TNF, IL-1B, and HLA-DRB1 contribute the most to classification. Higher bars indicate genes that have more influence on the model's prediction. Biological interpretation: IL-6, TNF, and IL-1B inflammatory cytokines involved in immune activation and joint inflammation. HLA-DRB1 is a known genetic risk factor for autoimmune diseases. The presence of these genes confirm that the model captures biologically meaningful patterns related to rheumatoid arthritis. A final conclusion, machine learning models can effectively classify rheumatoid arthritis using gene expression data. Among all the models, random forest performed the best due to its ability to capture complex gene interactions. The identified genes are biologically relevant and related to immune inflammatory pathways. The future scope: in the future, dimensionality reduction techniques like PCA can be applied. PCA, or principal component analysis, reduces the number of features while preserving important information. This can improve model performance and reduce computational complexities. Hyperparameter tuning can also improve SPM performance, and the model can be tested on larger datasets. Thank you, and we are ready for any questions.
Thank you so much, Group 3. Wonderful presentation from you. Thank you so much. So, question to you, Krishna. Mm, you somewhere you talked about the SVM not being a very suitable model for high dimensional data, right? Why do you think that? Why is SVM not very suitable for a high dimensional problem? Anybody from Group 3? Would you like to answer? Okay, let me put it this way. What kernel did you use when you performed the SVM classification? Sudiksha, Manaswini, Hasini, would you like to weigh in here? Guys, am I not audible?
We can hear you, Surya.
Yeah, we did not use linear kernel, sir. We used RBF kernel.
Okay,
Here, that's the reason we got it in a curve way.
that's the reason.
We got that linear graph. You can get it in straight because we have numerous genes, and it is better to use the RBF
Mm-hmm.
kernel for SPM instead of linear kernel if you have a lot of
Mm-hmm.
data. So use RBF.
Is it especially if you have a lot of data or if you have non-linear data, or rather if you have high
Non-linear
dimensional
data.
data? Okay, okay, no worries.
Non-linear.
Okay, no worries. Couple of suggestions here again, just like groups 1 and 2, and this is something that I am seeing is a trend across all of you guys. So this is not something which is acceptable, guys, because I have told you this every time. Okay, accuracies, precision, F1 score, each and every one of these metrics should be mentioned with mean plus minus standard deviation. Okay, if you are not doing that, your results are not admissible. Okay, so let me ask you this, Group 3, did you guys do any form of cross validation?
No, sir, we did not do any cross validation.
Okay, you should. No machine
Yeah,
learning
if we did
analysis.
not, we would have gotten the results of mean standard deviation.
No machine learning analysis is complete without cross validation. Your results don't mean anything if you don't do cross validation. Okay, they only mean that this is something that is a possibility using the data you have. Okay, but it cannot be something that can go in your report. if you have not done cross validation. Number one. Number two, did you guys do any form of hyperparameter tuning? Because you guys used what all models did you use? SVM, random forest, and
And logistic regression.
logistic regression. Okay. So SVM and logistic regression is fine. I guess you guys did not really do a lot of hyperparameter tuning there, which you could have, by the way. In even in case of logistic regression, you could have worked with the penalties, L1 or L2 penalties. Might give you some interesting results. What about random forest? Did you guys do any form of hyperparameter tuning? And if so, did you guys actually pay attention to the pruning parameters that we had discussed? Because that might actually help you prevent overfitting. Did you guys do any hyperparameter tuning using grid search CV or randomized search CV?
No, sir.
Okay.
No, sir.
Okay. No worries, no worries. These are just, don't take this the wrong way, okay? I am just pointing these things out to you because I feel these are things you guys should absolutely do. Okay, so please revisit this. You guys will always have the codes, you guys will always have the data to revisit this and do these things and see if any of the results changes. Even if it does not change, it works as a good practice for you guys. Okay. Another thing is, I don't see any references, so that is a very big red flag when you're presenting to somebody and you're claiming that the genes or the markers that you have got, they actually serve some purpose biologically. You should have literature backing them. Okay, I'm sure you guys would have gone through some literature. Okay, so please always put the references. Okay, and at the bottom of the slide where you're mentioning that particular statement. Okay, not at the end. Okay, so work on that as well. Apart from that, you guys have done a very good job. You guys did well with respect to the analysis. This is the first time you guys are doing any form of machine learning, and I think it's a valiant effort. So all the best to you guys, and thank you so much for this presentation. Thank you, Group 3.
Thank you, sir.
Thank you, sir.
Thank you, Suryadev, and thank you, Group 3. Moving forward, Group 4, Nuseva,
Yes, ma'am.
please start your presentation.
Please let me know if you can see my screen.
Yes, it's visible.
Good afternoon, sir. I am Nusaiba from Group 4, and I am going to be presenting our mini project topic, machine learning based classification of asthma via gene expression profiling. So, our goal here was to use gene expression data alongside machine learning to classify asthma patients accurately and identify the key genes that drive the disease. This is the overview of the presentation that I will be following. So, asthma is a chronic respiratory disease that is characterized by airway inflammation and mucus production. So, traditionally, it is diagnosed using symptoms and lung function tests. But, however, since asthma is a heterogeneous disease, meaning that different patients may have different molecular causes, um, this is where our role of genomics come in. By studying each gene expression, we can observe which genes are more or less active in asthma compared to a healthy individual person. So our objective here was to use machine learning to classify asthma versus healthy samples and identify the important biomarkers. So we worked with a gene expression data set that contained 48 samples, and each sample had thousands of gene expression values, making the data high dimensional. So our workflow consisted of pre-processing, that is data cleaning and normalization, followed by label encoding, where asthma was labeled as 1 and control as 0, and feature scaling to normalize the gene expression levels. And we trained three machine learning models. So logistic regression, that is a linear baseline, support vector machine, SVM, that is a maximum margin classifier, and random forest, that is a nonlinear ensemble model. So we compared models on the basis of accuracy, precision, FN score, and AUC. So logistic regression achieved about 71% accuracy, SPM performing slightly better at a 73, but random forest clearly outperformed both, achieving 98% accuracy and an AUC of 1. So this suggests that asthma gene patterns are nonlinear and complex, which random forests can capture better as compared to the other two models. So for the confusion matrix shows how well a classification model is performing. So over here, logistic regression model, it classified 34 out of 48 samples with 7 false positives and 7 false negatives. So this means it misses complex gene interactions. And SVM had a slightly better improvement than logistic regression. It classified fewer false negatives, meaning slightly better asthma detection. Now, for random forest model, um, it correctly classified 47 out of 48 samples with zero false positive. No healthy individuals were labeled asthmatic, and only one false negative. So, clinically, reducing false negatives is very important. Now, moving on to the evaluation. This is where we use ROC curves. So each point on the ROC curve represents one threshold and one confusion matrix, and an ROC curve is created by testing many different classification thresholds. So instead of evaluating the model at just one cutoff, ROC shows performances across all possible thresholds. So the ideal model lies at the top left corner here, which is random forest for us. And okay, so high true positive rate that is detecting asthma, and low false positive rate that is not misclassifying healthy people. So you've got AUC, that is area under the curve, that summarizes the ROC curve into a single number. So higher the AUC, better the model at across most thresholds. So in our results, logistic regressions and SVM had an AUC of 0.807 and random forest at 1. So over here, ROC helps choose the best threshold for a model, and AUC helps choose the best model. So because random forest had the highest AUC, it is classified best for our data set. So high performance is good, but in biology we need to explain how, why the model predicts asthma. So that's where SHAP comes in. So SHAP quantifies how much each gene contributes to the prediction. So in the SHAP plot, each dot is equals to one sample. A red dot is high gene expression, blue is low gene expression, and dots to the left push prediction towards asthma, that is positive SHAP, and dots to the left, right, no, that dots to the right, positive SHAP, and dots to the left is negative SHAP that pushes towards control. So this means SHAP tells us both which genes are important and whether high or low expression drives asthma. So for example, IL-3 appears at the top, and when IL-13 appears at the top, and when IL-13 is expressed highly, SHAP values are positive, strongly pushing prediction towards asthma. So this matches the biology where interleukin 13 drives airway inflammation and mucus production. So SHAP confirms our model is learning real biological mechanisms and not just random patterns. Okay, so for this plot shows feature importance for logistic regression, where the y-axis is the coefficient value, and the x-axis is the coefficient value, and the y-axis are the genes. So larger the magnitude, the more important the gene is. So positive coefficients push towards asthma, and again negative towards control. So when with this simple linear model, genes like interleukin 5, 13, 4, and TSLP, they appear highly influential. So these are the classic inflammatory cytokines that are linked to asthma. Now, this is the same idea, but for the SVM model. So again, genes like IL-5 and TSLP show strong influence. So the most important point is consistency across all models. So all highlight similar type 2 inflammatory genes. So this consistency increases confidence that these genes are truly involved in asthma biology. So for biological interpretation, for Th2 signatures, we've got core for inflammatory genes, interleukin 4, 5, 13, and TSLP. So these drive the mucus production ability modeling, and for genetic susceptibility for ORMDL3, this is frequently cited as a top asthma susceptibility gene. So our model correctly identified its high predictive value, and these references back up our interpretation. So in this table here, we've got the gene and the biological role it plays and the evidence across the models that we observed and what significance does it have in asthma. So together, these genes show that a single factor does not cause asthma, but by a network of immune and epithelial pathways driving the chronic airway inflammation. So to conclude, the best model for our dataset was random forest, which captured the complex nonlinear relationships in gene expression, and the model successfully identified a strong type 2 inflammatory signatures as the primary differentiator for asthma. So across SVM and logistic regression and SHAP analysis, these models consistently identified type 2 inflammatory cytokines and epithelial immune regulators as the dominant biological drivers for asthma classification. So this approach supports future precision and in medicine and biomarker discovery in asthma. Thank you.
Thank you so much, Group 4. It was a brilliant presentation. Really enjoyed the presentation. Very well done. So, question for you, Group 4, is first of all, Random Forest was the best performing model that you have showcased here. So, I want to ask whether you guys did any form of regularization on Random Forest. Was any regularization performed? Any pruning or any uh, different using different learning rates? Anything of that sort?
So basically, we try to do like first with XGBoost, but so we made a script using XGBoost also. Like, but we got like overfitting. It was overfitting. So then we
Mhm.
tried validating it, but it was still giving us like overfitting data. Then we changed it to the random forest, and because of the time limit, we just went for the default parameters. So
Mhm.
we did not do like regularization. But
Okay, okay. Try out the regularization and try different parameters. And my idea would be, so see, this is asthma, right? The disease is asthma. It is not something like cancer or something which requires, I should say, as much attention, because even if you do end up missing a certain few cases, it's not going to be lethal for that person, mostly in most cases. Okay, so don't kind of so if you remember, whenever you're doing a randomized search CV or a grid search CV, you can optimize based on each of these metrics, right? So I would suggest you can optimize based on precision. Okay, and check out random forest in this case. Just see if you get any results which actually tell you something more. Okay, this
yes,
is just a
yes,
suggestion. Okay. Also, Nuseba, you mentioned that you guys did feature scaling. Can you please tell me what kind of feature scaling you guys did? And was it done for all the three models?
Sir, we did min-max scaling.
Okay, we did min-max scaling. And do
That
you know what?
transforms all the features to a range between 0 and 1.
Okay, okay. So you did min-max scaling. And did you try with and without min-max scaling or only with min-max scaling?
No, sir, we did it with min-max scaling. We didn't try
Oh,
it without that because like scaling is important. Is like what we studied because it brings all features on a similar numerical range.
mhm, mhm. Okay, no worries. So you did min-max scaling. Very good. You know what min-max scaling does. Very good. Apart from that, very well done. I really like the fact that you guys did that table wherein you listed all the marker genes and all the models that gave you the evidence of those genes. Right? I really like that table. That is a very good touch. Very well done. And apart from that, yeah, I'm pretty happy with what you guys did. So thank you so much, Group 4. It was an amazing presentation and all the best. Thank you so much.
Thank you,
thank you,
sir.
sir. Thank you, sir. Thank you, Suryadip, and thank you, Group 4. Moving forward, Group 5.
Yes,
Ram- Yeah, please start your presentation.
yes, ma'am. I'm trying to share my screen.
Yeah, please. You should be able to share.
Uh, just a second.
Your screen is visible.
Okay, good evening everyone. I'm Ramya, and today I'll be presenting our mini project titled Comparative Analysis of Machine Learning Models for Breast Cancer Classification Using Gene Expression Data. Uh, so basically, the main aim of this project is to classify breast cancer samples using gene expression data and compare the performance of the different machine learning models. We used three different machine learning models, namely SVM, Random Forest, and XGBoost. So there's the problem statement we got and the goal. Um, so I'll start with random forest model. It's an ensemble machine learning algorithm, which means it combines the predictions of multiple decision trees to make a final decision. And each decision tree is trained on random subset of samples and features. This randomness helps reduce overfitting and it improves generalization. Um, so there's the accuracy, precision, and F1 score, basically the performance metrics we got. Um, uh, there's the confusion matrix. Here we got 27 true positives and 1 false negative, 2 false positives, and 26 true negatives. Next, I'll discuss about the ROC curve we got. This basically is a plot against true positive rate and the false positive rates. The curve lies close towards the top left corner and the AUC value is approximately 0.5. Um, it means that the model has high sensitivity and specificity. Next, we have future importance plot. Here we can see the future importance plot generated by the random forest model. Basically, it highlights the genes that contribute most to the classification. Genes with higher importance values play a stronger role in distinguishing cancer from the control samples. The presence of the small number of the dominant genes, they suggest that the specific gene expression patterns are strongly associated with the breast cancer genes. Um, Next, we have random forest models. Um, Sorry. Yeah. Next, we have sharp base form plot. So here each dot represent red color. The red color indicates high gene expression, and the blue color indicates low gene expression. Genes at the top of the plot have the highest influence on the predictions. Um, so basically from this plot we can observe that the certain genes, when they're like highly expressed, significantly increase the probability of being classified as cancer, and vice versa. um, uh, Next, I'll explain the XGBoost model, which stands for Extreme Gradient Boosting. XGBoost is an ensemble learning method based on gradient boosting. It builds decision trees sequentially, where each tree uh, corrects the errors of the previous one. So there's a metric we got, and the accuracy is 98.21% and the precision is 100. Um, we have confusion matrix here. True negatives are 28, true positives are 27, false positives are 0, and false negatives are 1. So this shows that the model correctly classified almost all the samples, with only one cancer case missed. Next, we have ROC curve. ROC curve shows that the AUC value is approximately 0.98, almost near perfect discrimination. Also, um, next we have sharp base form plot. Each dot here represents one sample, and again the red indicates the high expression, blue indicates the low expression. It basically summarizes global gene importance. And then only a few genes could contribute significantly to predictions, while most of the genes have minimal impact. This improves interpretability and increases trust in the model predictions. Um, sorry. Next, I'm going to explain about SVM model. It basically works by finding an optimal boundary that separates different classes. It is suitably for high dimensional data like gene expression data sets. The model was trained using a 80-20 train split. Uh, this is the accuracy, precision, and F1 score we got. Next, we have ROC curve and ROC curve. So the AOC score was 0.61, and as AOC value greater than 0.5 indicates that the model performance is better than the random classification and has learned meaningful patterns from the data. There's the confusion matrix again. We have 16 true positives, 12 false negatives, 9 false positives, and 19 true negatives. The feature importance from the linear SVM identifies genes with the stronger influence on classifications. Variations in the expression of these genes may be associated with disease conditions, and they may act as potential biomarkers. However, overall performance was weaker compared to the tree-based models. So the biological interpretation was the machine learning models identify as a subset of the genes whose expression levels strongly influence the classification of breast cancer and normal samples. These important samples are generally associated with the cell cycle regulation, cell proliferation, DNA repair, and apoptosis, which are key biological processes involved in cancer development. In cancer samples, abnormal gene expression patterns indicate uncontrolled cell growth and altered regulatory mechanisms. This shows that the model are learning biologically meaningful gene expression signals rather than random patterns. So these are the references. Sorry, so we had to put it on the last slide. And coming to conclude, we compared 3 machine learning models for breast cancer classification using gene expression data. SVM model showed moderate performance. Random forest comparatively achieved high accuracy and good interpretability, and XGBoost outperformed all the models with neat, perfect classification. SHAP analysis also confirmed that the models relied on biologically relevant genes as ESR1, BRCA1, BRCA2, and TP53. Thank you, sir. That's all.
Thank you so much, Group 5. Wonderful presentation.
Thank you, sir.
So, please don't turn off your screen sharing. I wanted to ask a few questions. Could you
Okay, sir.
please share your screen? Yeah.
Yes, sir. Yes, sir.
Yeah, just open up the PPT. Yeah. So again, another thing, this is the 5th group that I'm asking today. Guys, did you guys do any form of cross validation?
No, sir, actually, we did not.
Okay. Please, guys, cross validation is very important. Okay. I guess, okay, maybe the thing is that you guys did not really have a lot of time on your hands, and it was kind of last minute. So I understand that you had to do it during your regular classes, but please let this be known that cross validation is not optional. Okay, you have to do it. Okay.
Okay, sir.
With that said, references again, it was in the end fine. Also, the references I did not find any. I as far as the references that you have mentioned, none of them link the genes you got to the disease you are studying. These are references with respect to the models. So they are okay with respect to references for your PPT. But what I mean by references is that references which actually tell us about the genes that you have got. So rather, I would have preferred some references that link genes like BRCA1, BRCA1, which we also say as BRCA1, or genes like ESR1 to link it with breast cancer, or references with respect to finding these in gene expression data sets to be highly regulated, right? Those are references that ideally you should have put here.
Okay.
I don't want to see references with respect to random forest or XGBoost because those are the techniques we have used. Okay. And number three is that could you take me to any of the SHAP plots?
Okay.
Any SHAP plot, yeah. Yeah,
Yes.
this is fine. So this is the SHAP plot for which model?
This is for XGBoost, sir.
Okay, it is for XGBoost. Okay, so here do you see ESR1 and BRCA1, the
Yes,
top
sir.
two genes? Yeah. So could you explain what is happening here?
Sir, so basically it showed that those genes are lowly expressed in the
Lowly expressed in the
machine model.
uh, machine learning model.
Yes, sir.
Okay. Others from group 5, Lasya, Harshita, Manaswini, Lakshmi, would you like to weigh in here?
I did not perform this model, but like for those two genes in particular, it's overly expressed in the disease model
Mm-hmm.
and least expressed in the negative controls.
Okay. And would people agree with Lasya, what they said? Ramya, do you agree with Lasya?
Yes, sir.
What about you, Harshita, Manaswini, and Lakshmi? Do you guys agree with Lasya?
Sir, the red dots represents the high feature value and the blue dots
Mm-hmm.
represents the low feature value, right? In
Mm-hmm.
BRAC1 gene, which is at top,
Mm-hmm.
the higher BRSA1 gene, which is
Mm-hmm.
represented in red dot, it
Mm-hmm.
it expressed the increase in cancer prediction, and the blue dot on the left side, which express the decrease in cancer prediction.
In other words, Harshita, would you say that the red dots are more characteristic of breast cancer and the blue dots are more characteristic of control? Would you agree to this?
Yes.
Is that kind of where you were trying to hint towards, which is exactly what Lasya said? So, Harshita, am I right in understanding
Yes,
what you're
sir.
trying to say? Okay. Okay. Thank you so much, Group 5. Wonderful presentation. Really happy with you guys. You guys did a great job. So keep it up and all the best for all your future endeavors with machine learning and biology. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, Group 5, and thank you, Suryadeep, again. Moving forward, Group 6, Naraka.
Yes, yes, ma'am. Pallavi will be sharing the screen and I'll be presenting it.
Sorry. I'm extremely sorry, I couldn't hear you properly.
Ma'am, can you hear me now?
Yes.
Ma'am, Pallavi will be the one sharing the screen and I'll be the one presenting it.
I didn't hear.
Can you hear me now, ma'am? Why am I not audible? Hello?
Yes, sir.
Am I not audible, ma'am?
You're audible, but try to hear my once again.
Oh.
Bhavni, can you try mute? I think it's a problem with the network because the voice is echoing.
Fine.
Let's all mute. Sarak, can you try speaking one?
Yes, sir, I'll try. Like, can you hear me now?
Yes, this is better. Please continue.
Okay, how about now, sir?
Previous was better.
Previous was better. Okay. Okay, is this better, sir? I
This
don't
is
know why
better.
it's happening. Okay. Pallavi, did you share the screen?
Pallavi is over here to share.
Now I don't
Now
have
I don't have
permission.
permission. Pallavi. Give me a minute, Pallavi.
Sure, ma'am.
Sure, ma'am. Yes, please
Yes,
try
please
now.
try now.
Thank you,
Thank you,
ma'am.
ma'am.
So can I start now?
Please start.
Please start. Okay. Good evening, sir. This is group 6. My group mates are Sadaf, Priya, Tanmay, Pallavi, and myself. Our project is colon cancer versus control using gene expression data. It focuses on classifying the colon cancer versus control sample using high dimensional gene expression data, applying machine learning models with proper evaluation and explainability. So the data set overview would be, like I said, we worked with a biomedical gene expression dataset, which was named as Group 6 colon cancer versus control. Each row here represents one biological sample, and each column represents the expression level of that very gene. The data set contains 284 samples and 19,298 columns. Out of these, 19,297 are gene expression features, and one is the target label. The target variable label has two classes. Yeah, let's move on. Pallavi. One represents the colon cancer and 0 represents control or healthy species. There are 142 cancer samples and 142 control samples. This makes the data perfectly balanced. Because the classes are perfectly balanced, we do not require any resampling technique. The pipeline we followed for our project was first we understood the data, then we did the EDA, which is your exploratory data analysis, then we did the pre-processing, then we did the model training, model evaluation, model explainability, comparison, and then finally conclusion. So EDA for gene expression data sets, traditional EDA, that is your histograms, it was just not possible for each feature. It would be not meaningful because of the extreme large number of genes. So therefore, our EDA, it basically focused on class distribution. These plots over here, as you can see, we focused mainly on class distribution. So over here, like we can see, there is on x-axis, there's your expression level, and on your y-axis, there is your frequency. So the higher curve over there, it represents that the expression for these is lower, and hence these could be showing the control ones or the undiseased one. And as we move forward, the curve gets broader, and this shows that it is a colon cancer, like it is the diseased one. Similarly, and then we had the correlation heat map, which shows the pairwise correlation between the top 10 most important genes. The color scale, we also have the color scale, that is your -1 to +1, and +1 could be like the perfect positive correlation. It's like both variables increase, and minus is your negative. It's the vice versa. We also have like the oncogen synergy, and that is your PIK3CA, and cross, like they show the indicate they are often expressed together. And then we have the opposing relationship as well. The red one indicates upregulation, like it shows higher expression, while the blue is the opposite of it. It shows lower expression, low expression. And then moving on, we have our pre-processing. Pre-processing is extremely important in high dimensional biomedical data that we are currently dealing with. So the first thing we did was your label encoding. We encoded control as 0 and condition as 1. This is basically required because the machine learning moderates on numerical features. And then we did the train test split, the 80% into training and 20% into test using stratification. And then we also focused on scaling, and we applied scaling only for logistic regression, which you'll see in the next slides. So logistic regression over here, what it is. Logistic regression, it's suitable for high dimensional data only when regularization is applied, and logistic regression was used as a baseline model. Over here, we experimented with multiple configurations. So like we tried without the solver first, and then without solver and without scaling, and then we moved on to your with standard scaler and with min-max scaler. We observed something very weird, I guess. That was that after applying standard scaler, it dropped without, like when we did it without solver, like just the default one, that is your LBB, LGBFS, and your L2 penalty, we got 0.98. But when we applied your standard scaler and min-max scaler, it significantly dropped. Okay. We also have your confusion matrix over here. That is basically a table, and it shows the number of correct and incorrect predictions made by the model for each class. So it was your 28, which is your true positive, like it was correctly predicted as cancer. Then it was your 1, which was your false negative. That means it was predicted as control, which is which is not right. And false, we also had false positive, which was your 0, and then true negative, which was your 28, which is good. Now, if we move on, no, go back, Pallavi. We did observe that, when we did not use the solvers, we dropped, like from our accuracy went from 0.98 to 0.5 something, previous one, Pallavi. And this basically, sir, after I researched, it said that like when our group mates researched, it was for the loud voice effect, that without scaling the genes with huge raw numbers, they dominate the model. And the logistic regression wasn't looking at the biology, it was just basically memorizing the loudest numbers. And then with scaling, it just dropped because now those 19,000 plus features was just as unimportant as the 10 important ones. So this indicates that regularization and solver choice dominate performance more than scaling in high dimensional genomic data sets. And now moving on to when we did apply the solver and solver as liblinear and then L1 penalty. Over here, we observe that Pallavi, the L1, yeah. The next slide, Pallavi. No, previous one, that's fine. When we applied the L1 penalty, what happened is that the 19,000 features, what it did was it like ignored all like majority 99% of them. So that's why the precision and everything we got was 1. Precision and ROC and everything. Yes. And then we have like the top 10 important genes. These genes show clear difference in expression levels between cancer and control samples. Like you can see, these are the top 10 important ones. We have PIK, 3CA, APC, and CROSS. These are the major important genes, and along with their coefficients. Next one, Pallavi, random, yeah. To ensure robustness, let's now move on to your random forest. What random forest basically, like we did this after logistic regression. And random forest works by, it builds many decision trees, and each tree looks at random subsets of genes, and then the final prediction is made by majority voting. Yeah. And why we chose this is because we wanted to reduce overfitting, and random forest does not require feature scaling. And here the reason for that would be like, because the splits are based on the threshold comparisons, and magnitude of values does not affect the decisions. We did the baseline random forest before tuning, baseline. Yeah. First, we trained the baseline random forest using the default parameters. We did not do any hyperparameter optimization, just to see how the model performs initially. So like you can see, we got the OOB score, we got accuracy precision 0.9 to 0.9, 0.98, 0. Like it was closer. It was honestly very good. And even the confusion matrix showed very few misclassifications. What we could interpret from this was that even without tuning, random forest exactly performs very well because the data set has a strong signal separating a cancer and control sample. Now we tried one with the hyperparameter tuning. Yes, the baseline performed good, but we wanted to see what it would happen with hyperparameter, because again, default parameters may not be like optimal. And over here, we used randomized search instead of grid search, because randomized, it's unlike grid search, does not perform all possible combination. It just does random combinations, and it's faster and more efficient, and it also works well. So this is why we used. Also here, sir, we did do the cross validation, that is your 5-fold, and yeah, 5-fold, and like the data was split into 5 equal parts. We trained on 4 parts and 1 part for test, and then we repeated it 5 times. So this was more efficient than grid search. Grid search. Then we moved on to your ROC. ROC, Pallavi. Yeah, this one, right? This plots true positive rates versus false positive rates. So what true positive rate basically means, it's like it's a sensitivity, and out of all the actual positive cases, how many were correctly identified? So what were identified rightly? And false positive rate, this out of all the negative cases, how many model were incorrectly labeled as positive? So over here we see that the ROC curve shows the performance of random forest. The x-axis is your false positive rate, and y-axis is your true. The curve rises sharply to the top left corner. Like we can see, it's rising sharply to the top left corner, and this indicates that the model achieves a very high true positive rate, and like it's almost zero false positive. The AOC one indicates near perfect discrimination, and like the one AUC, what does it mean? It's like a perfect separation between colon and cancer samples. Now let's move on to the top 10 features. Again, we did the same thing over here as well. These are the important genes that were modified by random forest. Let's move on to the XG one now. Yeah. XG. Pallavi. So XG boost is an ensemble learning model based on gradient boosting, where decision trees are built one after the other. Each new tree focuses on correcting the mistakes. And unlike random forest that was independent, like it built trees independently, XG builds them in a sequential error-driven manner. So XG boost achieved the perfect classification performance. And let's go on to the accuracy. Yeah, the accuracy, precision, RF, and all the things that we achieved here were one. This performance is like you can see, it is consistent with your random and your logistic regressions. This suggests that there is a strong separability between the colon cancer and your control samples. ROC, let's move on to ROC, Pallavi. The ROC over here is again the same thing like we saw in the random forest. It's again, there's a true positive rate on the Y and false on the x-axis, and there is this sharp increase over there, which includes that there is a high true positive rate with very fewer false positive. Yeah. Then so we have your SHAP over here. What exactly is SHAP? SHAP is your Shapley additive explanations. It is a model oh god, I can't pronounce this. Interpretability technique that explains how much each feature contributes to a model. In simple terms, we could just say that how well or like which genes influence the model's decision and in which direction. Yeah, keep it here, Pallavi. This SHAP summary over here.
Time up.
Oh, okay, so.
Just go to your conclusion.
Okay, okay. Yes, sir, we do. And in summary, like the colon cancer gene expression dataset exhibits your strong biological separability, leading to a near perfect classification across multiple models. XGBoost was selected for detailed analysis because it was robust and it was and our data was high dimensional. The SHAP results confirm that the predictions are driven biologically and they are also relevant, and while acknowledging the limitations exposed. Yes, sir, done.
Thank you.
Thank you.
For the presentation. So question now to you guys. So you mentioned that I love the fact that you guys did some EDA, although it was not necessary, but well done. You did some EDA. That's good. So one thing is you did the KDE and histogram plots. My question is, why did you not separate it with respect to the classes? So because you had the code, right?
Yes, sir, we did. So actually, at that point, you can say that that didn't click into our mind that we could have separated it. So we just went with what like the flow we knew.
Okay. No. Mute one. Yeah. Okay, so my advice to you is next time when you do EDA with respect to these gene expression data sets, do it based on the classes. Okay, like showed you, right? For blue and orange, if you remember, our plots were, and they showed clear, distinct separations, right, in the histograms. And this is something that I also observed when you guys did the EDA. A lot of you did not do the class-wise separation, and that actually uh, you actually tend to miss out on a lot of information when you do that. Okay, you ideally want to separate it by class and then look at the distributions, because that is where the differences will actually show up. Okay, with that said, Sarah, I also see that you had mentioned that you did the heat map of the top 10 most important genes. How exactly did you decide which are the top 10 most important genes?
Sure, again, there was like a code, and we have done this using the logistic regression one. So, like, we had a code, and then we just let the model do the decision making.
No, so the choice of the genes, that has to be made by you, right? What to put on the plot. Uh, who did this in your group? Maybe they would be able to explain better.
Actually, sir, we just, I don't know why we didn't try to do that, but we just went on with the regression, and we just did this plot. Unfortunately, we didn't use our own brains to decide which one is the best.
No, I'm saying that you are mentioning here that you did the heat map of the top 10 most important genes, right? And that is absolutely fine. But on what basis did you uh, choose that these are the top 10 most important? You, there must have been
As
some
in,
sort of
like,
logic behind it, right?
some are like overly expressed and some are less expressed, and this shows the correlation where some genes which cause cancer express overexpressed together, whereas some underexpressed.
So did you do an exhaustive heat map and then picked the ones which showed the highest correlation? Is that what you did? Or did you do feature
Unfortunately,
importance?
we did not. Uh,
Yeah, go ahead, go ahead.
we did not, sir. Our PC was not as good, and I don't know why it was slightly crashing. It was already kind of overheated, probably not because of the code, but I'm unsure. So we felt like just doing the logistic regression and then using it for the plot would be better. Um,
No, I get that you did it based on logistic regression. I'm asking, how did you figure out that these are the 10 genes that you want to showcase on the heat map? What was the logic behind choosing only these 10 genes? Who did this in your group? Maybe they would be able to give you a better idea.
So, like in the code when we were writing, I mean, I had researched it, and we had made a code in such a way that it would pick the top 10 important genes. And then we got this, and then like when we searched about these genes, they were shown to be important, and so like yeah, that's how we did it.
Okay, so you wrote the code in a certain way. I'm guessing, Sarah, could you please unmute? So sorry. Yeah. So I'm guessing the code that you wrote, you picked it from some external sources, right? You took help from some external sources. So did you actually understand why it picked these top 10 genes from the code? Because that is really important.
Yes, so like kind of because the model itself evaluated, right? So the model picked it, and then when we did try to find them on Google, like when I searched these genes, it did say that these have an impact, or like these are the major ones. So kind of like that.
So in other words, you use the logistic regression feature importance to pick these top 10 important genes. Is that correct?
Yes, yes, so that's right.
Okay. Okay. I hope you understood the code that you have used, right? You have not blindly used it. I hope that is because that is really important.
Yes, sir, I did. I mean, how much ever I could, like my group mates and I could, we did, and the remaining, we would try to like ask what's happening, and then yeah.
Okay. Okay. Very good. Group 6. Apart from that, so you guys also did the hyperparameter tuning, I suppose. Yes.
Yes, sir.
Uh, so whenever you are doing hyperparameter tuning, make sure you mention the best parameters. Okay. So whatever were the best parameters, whatever the best values of each parameter, you should always mention that. And when you get that best estimator object, basically, the idea is you should do a tenfold or a fivefold cross validation on the best estimator itself now. So the idea behind randomized search CV is to give you the best estimator. Once you have identified the best estimator given your data, you should use that best estimator to do the cross validation and then report the metrics in a mean plus minus standard deviation manner. Okay, is that understood?
Yes, sir. Yeah.
Okay, so please revisit that. I think that will give you a much clearer understanding. And also, I don't know if I missed it or not. Did you guys go through the actual functionalities of the genes and how it links to colon cancer?
Yes, sir, we did, and I like
Yeah.
the time limit was over. We did,
Okay.
so we did.
Okay. And so you mentioned down regulation and up regulation. So I believe this is in accordance to the sharp plots, right? Show me
Yes, sir,
any one of
sharp.
the sharp plots. And yeah, so KRAS, I can clearly see here. What do you see for KRAS, Sarah? Can you please give me a quick overview?
So what I see for KRAS is that in the left side, this side is the one that is for your undiseased, like it's the control one, and on your right side it is the your colon one, the cancer one, and the red one is your high feature value, that is your upregulation, and then your low feature value is your blue, and that is your less expression, less gene expression.
So what is associated with high expression of KRAS, control or colon cancer?
So I guess it would be your colon cancer.
Do other members of the group agree?
I think it's control.
You Pallavi thinks it's control. Sarah, do you agree with Pallavi? Or
Oh
would you
yeah.
like to? So
Okay,
think
sir.
about
So
it.
the red, okay, the pink one is on your left side. Yes, sir. It would be control. Yeah.
So KRAS would upregulated or downregulated in case of colon cancer, according to this plot.
Down-regulated in case of colon
Down.
cancer.
Yes, down, sir.
Correct. Okay. Okay. Thank you so much, Group 6. Very well done. Good presentation. Good that you guys also did some media, although it was not necessary, but I appreciate it that you guys did some media. Thank you so much. All the best for all your future endeavors with respect to machine learning and biology. And overall, very good job done. Thank you, Group 6.
Thank you, Suryadev. Thank you, Group 6. Moving forward, Group 7. Sadvedi is a presenter. Group 7. Can you please unmute yourself, Sadvedi?
Yes.
Thank you.
Good evening, everyone. We're Group 7, and we're here to give a presentation about machine learning based classification of project and cancer. Suppose the prostate disease is one of the most common cancers that affect the human beings, and it's usually categorized by swelling of the prostate glands or abdominal growth of the prostate glands. And since our data set is related to the prostate disease, it 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 compares the prostate disease patients with the healthy patients, which who do not have the disease. And the machine learning workflow is that first we load our data set, and then we separate the features and the target values, and it's mostly in the ratio of 80% and 20% when it comes to the training and testing sets when we split them. And then we perform the feature scaling and train multiple classification models and then evaluate them. And next, when we the models used for this machine learning is logistic regression, linear baseline model, support vector machine, non-linear classifier, and others. And so first, accuracy, precision, and reference scores are there, can be derived, and we got an accuracy of 0.79 and a precision of 0.00. And this is our ROC model, and this basically measures the ability to distinguish between prostate disease and the healthy patients. And where the higher ROC and AUC values indicate better classification capabilities. And coming to the confusion matrix or the SVM, it basically illustrates the true positives or the true negatives. The true positives are the ones which are correctly identified prostate diseases, and true negatives are correctly identified healthy controls. And the false positives and false negatives indicate miscalculations. So here the 39 is the true positives and 10 is the true negatives, and we had zero miscalculations. Then coming to our importance plot, these are the obtained from random forest model and displays the top 10 most influential genes. And then like we see here, the most important genes identified by the model are biologically relevant to prostate disease. And here, KLK3 or PCA, widely which is used as a biomarker for prostate cancer diagnosis, plays a central role in this prostate cell growth and is one of the most important biomarker in this. And then these are the rest other biomarkers which are responsible. And next, so yeah, in conclusion, machine learning models successfully classified prostate disease using gene expression data, and random forest achieved the best predictive performance, and to demonstrate the effectiveness of the machine learning approaches in genomic analysis. Thank you.
Thank you so much, Group 7. Could you please don't stop sharing? I wanted to ask a couple of questions. Group 7, Sadvedi, could you
Yes,
please share your
yeah.
screen? Yeah.
Yes.
Yeah. So, what all models did you say you tried out?
Sorry, sir.
What all machine learning models did you try out?
Yes, sir. We did the ROC AUC one and then the SVM one.
No, no, ROC AUC is a
Yeah, we did the SVM.
it's a metric. So which algorithms? I mean, so you used SVM. Apart from that, any other algorithm you have used?
Sir.
Sorry.
No, sir.
Okay, so you've used SVM. Okay, so you've used SVM. So let me ask you this: which kernel did you use for SVM? Group 7, anybody would like to answer? Pallavi or Sahiti?
Hello, sir. We've used SVM, random forest, and logistic regression. And the kernel we use for the SVM is like RBF kernel.
Okay. Pallavi, I did not really see the results of the other two. I only saw the results of, I think, only one of the models. Was that by choice, or did you guys
Um,
miss out
no,
on
sir,
the
we
other?
didn't have enough time. Like we've done all this process in the last minute, so maybe our mistake.
Okay, okay, okay. Okay, no worries, Pallavi, no worries, group 7. So my advice to you guys is, if you've paid attention throughout this session, right? Some basic suggestions is, try out different models, try out regularization parameters, hyperparameter tuning, as well as very important cross validation. Okay,
Okay, sir.
so please do revisit this
Okay.
and do it as and when you find some time. Okay, this is only
Yes,
for
sir.
your practice. Okay,
Yes, sir.
okay, no worries. Thank you so much, group 7. Very
Thank
good
you,
effort.
sir.
Thank you, and well done, very well done, group 7. Thank you.
Thank you, sir.
Thank you, Suryadev. Thank you, Group 7. Group 8, Shivani.
Hello, ma'am.
Yeah, please start your presentation.
Yeah, ma'am. Sindhura will be present the screen and I will explain the PPT.
Give me a second. Yes. Sindhura, can you please try sharing your screen?
Yes, ma'am. Just a minute.
Sindhura, your screen is visible. Can you please change to slideshow? Yes, let's proceed.
Yeah. Good evening, everyone. Today we are presenting our project titled Machine Learning Based Classification on Parkinson's Disease using the Gene Expression Data. The project was done by Group 8 and I'm Shivani along with my group members. And next slide. Yeah, the introduction. Parkinson's disease is a progressive neurodegenerative degenerative disorder. It mainly affects movement and occurs due to the loss of dopaminergic neurons in the brain. And early diagnosis of Parkinson's disease is very important. And gene expression analysis helps us understand changes at the molecular level. And machine learning helps us classify patients accurately. So in this project, we combine biology and machine learning. Um, our motivation is that gene expression data can identify important molecular signatures. Machine learning can help in classifying diseases and healthy samples. It also helps us find important genes and genes related to Parkinson's disease. To classify Parkinson's disease versus healthy control using the gene expression data and identify biologically relevant gene is the objective of this objective. And we used a gene expression dataset that compares Parkinson's disease patients with healthy controls. In this dataset, each row represents a biological sample, each column represents gene expression values. The target label is 0 for healthy control and 1 for the Parkinson's disease. Our goal is to train machine learning models using this data. Machine learning workflow. Our workflow includes loading the data set and separating features and target labels, and as well as splitting the data into training and testing sets, and scaling the features, training multiple machine learning models, and evaluating performance and identifying important genes. And this terminal outputs, this slide shows terminal output which confirms that the models are trained successfully and evaluation matrix were generated. This output validates that our code executed without errors. As well as train test splits, we split the dataset into 80% of training data and 20% of testing data. We used stratified splitting to maintain class balance. This helps prevent data leakage and ensure fair evaluation. We trained three machine learning models. First one is the logistic regression, which is a simple linear baseline model, and the next one is the random forest classifier, which works well for high dimensional gene data, and third one is the support vector machine, which captures the complex gene expression patterns. These models helps us compare different approaches. And the performance matrix, we evaluated the models using the accuracy, precision and F1 score. The value shows the moderate but meaningful classification performance. These matrix helps us understand how well the model predicts Parkinson's disease. And the ROC AUC analysis, where ROC AUC analysis measures how well the model separates Parkinson's disease from healthy samples. A higher value means better classification. Among all models, Random Forest achieved the highest ROC AUC score, making it the best performing model. The confusion matrix shows true positive and true negatives, false positive and false negatives. This helps us understand where the models makes the correct and incorrect predictions. The random forest model shows the balanced performance. The feature importance, we extracted feature importance from random forest model. Only the top 10 genes are displayed. These genes contribute the most to disease classification. The important genes identified are biologically relevant. SNCA, which is associated with alpha synucleic aggregation, and then LRRK2, which is linked to the familiar Parkinson's disease, and PINK1 involving mitochondrial function, and PRAK7 protects neurons from oxidative stress. These genes are well known in Parkinson's disease research. To conclude, machine learning successfully classified Parkinson's disease. Random forest performed the best. Important genes matched known Parkinson's biology. This shows usefulness of machine learning in genomic disease analysis. And thank you for listening. We will happy to answer any question.
Thank you so much, Blue Kate. Wonderful presentation. So my question to you guys is, so you guys also did random forest, right?
Yes, sir.
Were you able to do any sort of sharp plot?
No, sir, we couldn't.
Okay. Okay. So the sharp plot could have actually told you much better in terms of what is being upregulated and what is being downregulated. That could have been nice. The other thing is, so you mentioned that you guys have done 3 models. but I think you guys showed the metrics of the three models with respect to the terminal outputs. So I guess again it is a last minute thing that you guys did. That is why you could not like properly put it in a table. But which of the models performed the best out of the three?
So random forest.
Okay, and what were the numbers? Because I did not really get it.
Just a second.
Oh, sir, actually, this graph shows the most important genes used by the random forest model for prediction.
Which one? This is the confusion matrix you have also. I remember the plot, the feature importance. Is that the one you're talking about?
SNCA has the highest importance, meaning it plays the most significant role in the prediction.
Show that to me on the slide. I remember you guys have a slide, right, for the feature importance. Okay, group 8, no worries. I think you guys were a bit tight on schedule here. So no worries. Just as I said for group 7 as well. Okay, the general instructions apply to you all as well. Okay, do revisit this assignment and make use of the codes that I have already provided to try to understand. If you have doubts, feel free to reach out to Michael. They can always connect you with me and I can help you out. Okay, so no worries with respect to that. All in all, good job done, I should say. Even with a very busy schedule, you guys have pulled off a really admirable, really commendable project. So a big round of applause to every single person here, every group. I'm really impressed with how far you have come in the past 2, 3 weeks or so. Okay, I hope this internship has given you guys a very clear idea as to how you can deal with gene expression data in different fields, right? So no matter what field or no matter what disease area you're researching, The techniques all remain the same. It's just about tweaking them and tuning them as per your disease field or research. So I hope that is a learning you will take away from this internship, this entire 30 or 60-hour program that you guys have gone through. So first of all, a big round of applause to everybody who has sat through this entire 60-hour program. Your dedication is really commendable, and I'm really proud of all of you. Machine learning is not something you learn overnight. It comes with practice. So just to give you guys some context, I had an entire year of machine learning when I was learning it, when I was studying it in college. Okay, it also consisted of other high level concepts, but still, whatever I showed you guys is a very small subset of what machine learning in biology is all about. There is so much more to it. So I hope this forms a very nice baseline or a very nice foundation for you guys to explore further and dive deeper into the world of machine learning and how it ties up with biology. Okay. So wishing you all the best for all your future endeavors. And I hope our paths cross again once in the future. And I hope I get to spend some more time with all of you because I really enjoyed this entire time. Right? I hope you guys have a similar. I hope you guys share a very similar feeling with me. It was a really wonderful journey for me. Thank you all for having me, and thank you everybody for listening to me very patiently. And very, very, very hearty congratulations to all of you for learning what you have done in the last 15 days and putting that to application and actually pulling this project off. Consider yourselves to be very able. This is not something everybody can do, so make very well. Okay. Thank you, everyone, and wishing you all the best. Thank you so much.
Thank you, Suryadev, for your valuable words. And before we conclude, I guess there was few confusion regarding names in Part A submission. Would you like to clarify it now?
Yeah, the one with Praneeth we already clarified. The other one was, I think, Sai Krishna. Are you here in the call, Sai Krishna? Yes.
Sai Krishna is here.
Sai Krishna, did you turn in finally? Because I believe you had submitted. I mean, you had uploaded the file, but you forgot to turn it in. I think. Can you confirm?
And also there was a one named Charan.
Yeah. So who is Charan, guys? Charan P. I see a submission here by their name. Can anybody please confirm who that is?
Any Charan here?
Ma'am, accidentally I did, ma'am. This is Sai Krishna.
Okay. You submitted name of Charan?
So, sir, my brother mail. So accidentally I'm submitted,
Okay.
but I submitted my one and I turn it on.
Got it. Yeah. So
Okay,
is it clear now?
So Sai Krishna, yours should I evaluate the one under Charan or should I evaluate the one under let me see here, Sai Krishna Panchagiri? Which one would
Sai
you want?
Krishna. Sai Krishna.
Sai Krishna Panchagiri, right? So you
Yes,
have not turned it in. I don't think you can anymore, but okay, no worries. I will evaluate it based on that. Okay.
Okay, thank you.
Same for you, Sindhura. You also, I think, have not been able to turn it in. So
Yes, sir.
yeah, I will evaluate it based on what you have uploaded. Okay.
I'll try again once.
If you can, just try to turn it in. If not, no worries. I see.
I don't think so that will work, Suryadev,
Because the deadline is already gone. I don't think you can turn it in. No worries. I will evaluate you based on whatever you have uploaded. Let me just check once if I'm able to open it. Just give me a sec. Yeah, I'm able to open your upload. So fine. I will evaluate you based
Okay,
on
sir.
that.
Thank you, sir.
Apart from that, everybody else has turned it in, apart from the people I have mentioned in the Telegram channel. So those the names that I have mentioned in the Telegram channel, you guys won't be evaluated for part A, and your respective group overalls will go down because of it. Okay. just information that I wanted to give you guys. So please bear that in. Okay, Bhavni, I think with respect to the group evaluation, we are done.
Yes.
We can proceed with the next steps.
Fine. Thank you, Suryadev. And I'd like to thank all the participants for your presentations today. And we truly appreciate your effort you have taken to prepare and demonstrate it right now. So it's well done, guys. So regarding the assessment results, we will require some time for evaluation. So assessment result will be posted in a Telegram forum. So please keep an eye on the updates. Is that clear? Please give thumbs up. Fine. So, as usual, before we conclude, I'm opening the floor for you guys. If you have any feedback or if you have any suggestions about our program, please let us know that. Any feedback from anyone of you?
Or if
We
you
just want to know your experience throughout this program. We think we might have already attended few programs before us, but we just want to know what experience you got from this program. Yes, Suryadev, you was about to say something, right?
Yeah, I mean, feedback is always welcome. So we would like to know, especially where we could do better, because if you guys have done some other programs as well, we would definitely like to know where you guys think we could have done a little bit better, and we will definitely work.
If you couldn't unmute and speak up, no issues, please post it in the chat. Or we have also shared the feedback form for you. You can also feel free to post it there. Anything is fine. Anyone would like to unmute now? Is Saida, you was about to say something?
Yes, ma'am. Actually, I texted in the chat. I wanted to ask how can we connect with Suryadeep sir after this internship, like if in case we want to discuss about any projects that we do in the domain of machine learning.
If you want to connect with him, you can post it in Telegram forum. That forum will be active and it's for lifetime access, so you can post it and you can notify him. And I'm also sharing you the another email address where you can connect with Suryadev. If you have any doubts specific to him, you can just write a mail here. We will forward it to Suryadev and he will get connected. One more time, Telegram forum will be active always. And I hope Suryadev will be there to respond it.
Absolutely. You can reach out to me. Telegram is one way. Another way is you can reach out to Cyberman as well. The email shared, they will relay the information to me, and we can always discuss any ideas that you have. I can always help.
Yes. If you want to carry out any project in ML, and if you wish Suryadev want to be your instructor, definitely we can do that. So we can reach us. All right. I think we came to conclusion. Suryadev, do you want to say anything to them apart from what you instructed before?
Not as such. I think I already gave my 2 cents on the internship. I'm really happy for this, and I wish you guys all the best. I wish you all do very well, and congratulations to all of you for getting so far, right? And it will only get better. Okay. Okay. So,
Thank you.
all the best to all of you. Thank you so much.
Yeah. So before we conclude the session, there's a quick update regarding your quiz and certificate. We are yet to receive from final confirmation from your department. So once we get the confirmation, we will inform you about the quiz date. Tentatively, the quiz is planned for second week of February. So until then, we kindly request you to prepare. Please do review the shared study materials and recording. Only from that, the questions will be asked. And also at this moment, I would like to take my extend sincere thank you to Tutor Suryadev for his excellent guidance and effort throughout this program. I think his clear explanation, interactive approach, and also continuous support truly made this session an engaging one. So we greatly appreciate your dedication and commitment, Suryadev. Thank you once again.
Thank you so much, Rodney. Thank you so much to Cyberman as well. And thank you so much to all of you. I really enjoyed this, and I hope our paths cross once again.
Thank you. With this, we came to end of today's session, and it was great pleasure working with you all, guys. We have made truly valuable session, I guess, and we hope this will be taken forward in your life. And we wish you a bright and successful future. And we're looking forward connecting with you all once again through another interesting program in future and the possibilities of collaborative projects as well. Thank you all once again. All the very best, everyone.